We know that researchers has discovered many life-saving drugs for human being. But suppose you wanted to discover a painkiller. At the end of the research, what you discovered has no analgesic properties. But the drug can be used to treat fever. This is serendipity. Discovering something by chance is called serendipity. There are many life-saving drugs that scientists discover accidentally during research. Let us know about these life-saving medicines. Penicillin. In 1928, Alexander Fleming was researching influenza. In 1918, an influenza pandemic, known as the Spanish flu, killed countless people. The bacteria responsible for influenza is Staphylococcus. In his laboratory at St. Mary's Hospital, Fleming was studying Staphylococcus bacteria in petri dishes. After work, he inadvertently left the petri dish on the windowsill. When Fleming returned to work after a month's leave, he saw mold growing in a petri dish of Staphylococcus bacteria. Staphylococcus bacteria did not grow in places where mold grew. He realized that mold contained a substance that could inhibit the growth of bacteria. This mold was Penicillium notatum. He named the substance that inhibited the growth of flaming bacteria as penicillin. Alexander Fleming won the Nobel Prize in 1945 for the discovery of the breakthrough antibiotic penicillin. Antibiotics in the treatment of ulcers. Barry Marshall and his colleague Robin Warren discovered the presence of the bacterium Helicobacter pylori while examining biopsies collected from ulcer patients. In 1980, these two doctors claimed that Helicobacter pylori was responsible for stomach ulcers. To prove this, Marshall collected Helicobacter pylori from a patient's stomach and injected it into his own body. Shortly thereafter, Marshall developed symptoms of an ulcer, which cleared up with antibiotics. In recognition of this discovery, Marshall and Warren were awarded the Nobel Prize in Medicine in 2005. Lithium. After World War II, psychiatrist John Cade injected urine collected from healthy people and mentally ill patients into the abdomen of guinea pigs. Guinea pigs injected with the urine of a mentally ill person died faster than guinea pigs injected with the urine of a healthy person. John Cade discovered that excess uric acid was responsible for the rapid death of guinea pigs. As a result, he concluded that patients suffering from mental illness have excess uric acid in their blood. John Cade added lithium solution to increase the solubility of uric acid. After inserting it, he found that the guinea pigs were calm instead of excited. Cade also administered lithium to his own body to prove whether it was safe for humans. Since then, he started using lithium to treat various mental disorders. Chlorpromazine. On December 11, 1950, Paul Charpentier synthesized chlorpromazine. In May 1951, a well-known pharmaceutical company in France at the time exposed chlorpromazine for clinical investigation as a potential general anesthetic. A clinical investigation conducted in February 1952 showed that chlorpromazine does not cause unconsciousness, but it causes a feeling of sleepiness and disinterest in the surroundings. This led to the use of chlorpromazine in psychiatry. Imipramine. Because of its structural similarity to chlorpromazine, Swiss pharmacist Ronald Kuhn suggested that imipramine could be used to treat schizophrenia. But the drug has not been found to be effective in treating this disease. Kuhn applied it to a patient suffering from severe depression before sending the drug back to the manufacturing company. He found that the patient's depressive symptoms were subsiding. Thus Kuhn discovered the antidepressant effect of imipramine on January 18, 1956. Chloral hydrate. Justus von Liebig, a German professor of chemistry, developed chloral in 1832. Pharmacology professor Otto Liebreich developed chloral hydrate for medicinal use 37 years later in 1869. Liebreich hypothesized that chloral hydrate contained a substance that was converted to chloroform in the body and would induce sleep in the same way as chloroform. But the breakdown of chloral hydrate in the human body shows sleep-inducing effects even though no chloroform is produced. Chloral hydrate was the first synthetically produced credible sedative that is still in use today. Nitrous oxide. Joseph Priestley was famous for separating oxygen and carbon monoxide gases. In 1772, he isolated another important gas called nitrous oxide. As the gas enters Priestley's body during inhalation, his body becomes calm and numb. After realizing the effectiveness of nitrous oxide as an anesthetic, its use in medicine began. Warfarin. In the early 1920s, veterinarians noticed that some cattle were bleeding profusely after injury or surgery. They discovered that the cattle ate the decaying clover. 20 years later, researchers developed the anticoagulant agent warfarin from the clover plant. Although discovered by chance, the efforts and wisdom of scientists were behind the discovery of these life-saving drugs. Therefore, many scientists have received various awards in recognition of their efforts and wisdom. Who knows, maybe in the future a medicine will be accidentally discovered which will save the lives of countless people. That's all for today. See you in another video. If you enjoy the video and found it informative, then please like the video and let us know your opinion in the comment box. Never forget to subscribe the channel and press the bell icon next to it. Share the video if we earn it. Thanks for watching.